Welcome, everyone. On behalf of Cambridge Health Tech Institute's Global Web Symposia and our sponsor for today's event, Visual Sonics, I'd like to welcome you to latest in vivo imaging to reduce clinical attrition rates. My name is Elizabeth Lamb, and I'll be your host for today's event. At this time, I'd like to introduce our moderator for today's event. She is Dr. Meenalina Lakshma, Applications Manager, Fujifilm Visual Sonics. Meena, welcome. The presenter ball is yours.
Hi, thank you, Elizabeth. That was a great introduction. I want to begin this by thanking you all participants for attending this webinar. We are very excited, partly because we're going to share with you the expertise of this curated panel of guest speakers. They bring their expertise in drug discovery and development, but more importantly, focusing on the use of the vivo imaging systems. So to put their presentation in context, I direct your attention to the next slide. The topic being that this is the latest in vivo imaging to reduce clinical attrition rates is very important, particularly when you focus on drug development in the oncology field. But it's not limited only to that field. For your perusal, I have provided you with a pathway that you see out there from a Nature Review publication last year. And this just illustrates the discovery and development pathway that is so elaborate in bringing drugs to market. Now, depending on your role within your company, I hope you see value in what our guest speakers have to talk with the use of the vivo imaging systems to advance their own research in drug safety, toxicity, efficacy, and also in the management of data that allows us to take it from your preclinical strategy into the clinical space. Without further ado, I want to make it clear that our overall goal for this webinar is to help enhance your preclinical strategy by providing you an opportunity to learn about how the vivo imaging systems have been used in these three points. So I'm going to pass on the presentation to our first speaker, and I'll put on his biography and the title right here. This will be Andrew Needles, our Senior Manager at Fujifilm Visual Sonics, who will be presenting on the introduction to high-frequency ultrasound. Andrew?
Great. Thank you very much, Meena. It's a pleasure to be here today. Thanks to all the attendees for taking the time to join in. So my portion of today's presentation will really focus on high-frequency ultrasound, how it can be used as an advanced imaging technique for drug development. I'll talk a little bit about a term called micro-ultrasound, which is actually another more specific term of high-frequency ultrasound. Show a few brief applications in terms of toxicity and efficacy, but I really will leave the details and much more, I would say, in-depth examples to our other panelists who have joined us today. And I'll talk a little bit about the importance of data management and some new products that we've been developing at Visual Sonics towards this end. Meena introduced this drug development pathway, as you can see on the slide here. I apologize, I don't think the image is showing under toxicity, but there will be plenty of examples in the coming slides. What I was showing in this example was a mouse heart, the parasternal short axis. This is a very, very well used technique for looking at toxicity effects, side effects through drug development. And one of our panelists, John Hayen from Pfizer today, will elaborate more on this topic. That's one side of it, but how can the vivo imaging systems also be used in terms of efficacy? Looking at tumor burden, tumor growth, here we're looking at very small prepalpable tumors in the mammary fat pad. This is on the bottom right portion of your screen. You can see that these tumors are extremely small, just under about 0.5 millimeter in diameter. So again, prepalpable tumors. This is the very first stage. The progression of these tumors or subsequent treatment can obviously be tracked with the system, and this is something that we'll delve into today with more examples, both in terms of tumor size and volume, functional imaging and blood flow, as well as perfusion kinetics. Just to give you a little bit of background on the technology for those that may not be familiar, I think many are perhaps familiar with conventional lower frequency ultrasound in the 3 to 15 megahertz range. You can see here an image of the human fetus We've given the honeydew melon here as sort of the size to give you a sense of the scale that we're working on with conventional ultrasound. What we've done and pioneered at Visual Sonics is really scaling the frequency to much higher frequencies, above 30 megahertz. And as a result, this really takes us into a much higher resolution imaging regime. You can see an analogous image from the mouse fetus in this case, looking at the developing fetus inside the mother. Here we're now on the scale of the coffee bean, which is much, much smaller, on an order of approximately 5 to 10 times smaller structures, and you can see the resulting resolution. Here we're getting 30 micron resolution as compared to something in the 300 micron resolution conventional case. So again, high frequency is really the technical term for what we've engineered at Visual Sonics. Micro-ultrasound is the more widely used term. It's a micro form of ultrasound. And what we can do is look at very small structures in small animals when looking at preclinical studies. Just to give you a sense of how the imaging is done, a schematic here, a transducer generates ultrasound waves. It propagates into the animal, in this case, using some coupling gel. The reflected sound comes back, and what we're really looking at is a map of variations in the tissue density and the different reflection boundaries. So again, we're looking at a developing mouse embryo in utero here, and we can see some structures such as the developing heart. We can actually see a ventricle in the brain, the nose, other fine structures. So again, the scale here is on the order of 30 microns. It just gives you a sense of how we can visualize in vivo many of these structures within the living organism. A few other benefits of ultrasound: it is high resolution, and unlike other techniques, we can actually maintain this resolution at depth as we go throughout the image. It is real time, so you can get all of this information live. There's no post-processing involved to get the actual image. As I mentioned, it's non-invasive, it's in vivo, and I should also add, it's not on this slide, but it's actually a very safe form of imaging. There's no ionizing radiation or other harmful effects that we would find perhaps with other imaging techniques. So how do we actually collect these images? What does the product look like? This is our new flagship system, our Vivo 3100. Here you can see an image of the system. It's a platform that's very easy to use, intuitive, touch-based display, lots of onboard learning and tutorials for all of these different studies that you may be interested in doing. There's how-to guides, imaging guides, and educational videos on board. We've also engineered a number of new transducers to get even better sensitivity and depth and image quality. So I won't spend too much time going into the details of all the system, but I want to make you aware of it, that this is actually what the micro-ultrasound system looks like. If you'd like more information, I encourage you to please go onto our website, look at all of the product information, or please contact us if you have other questions about the technology itself. I'd like to shift now just away from, just the system, but into more the applications and how this can actually be used. Specifically looking at efficacy, this was a study done using sunitinib, looking at tumor volumes, and we can see on the left side control versus on the right the sunitinib-treated tumors. These tumor volumes are all generated using analysis tools with the Vivo imaging systems. These volumes can be traced and calculated, and we can see quite clearly that as the treatment takes its course, the sunitinib-treated volumes are reduced relative to the control untreated animals. So this is a very, I would say, robust and well-used technique with the technology. All of these volumes are measured using 3D ultrasound, and it is really a very good way of assessing the efficacy of the different treatments that you may be applying in your studies. We can take this another step and actually start looking at blood flow. This is using a technique called power Doppler. Here we're looking at day 1 and day 4. No treatment in this case, but just looking at the progression and proliferation of the vasculature within this tumor. And we can do calculations of percent vascularity, or PV, as it's shown here, indicating that there is an increase in blood flow. We'll see more examples of this actually taking a look at contrast agents as well with our guest speaker from Regeneron today, who will be talking about the role of micro-ultrasound contrast-enhanced imaging for looking at blood flow and perfusion kinetics in terms of drug efficacy. Moving now to the toxicity piece, this is, again, part of that same study with sunitinib, actually looking at cardiac function, and we can see that there is actually a decrease, mainly due to heart rate, in this case looking at cardiac output. All of these measurement tools you will find on the vivo platforms. The measurements can be done, they can be reported, and as we'll see more examples of later, how this data can be interpreted, as well as how it can then be all that data brought together and managed for very large-scale studies, looking at large treatment groups, hundreds, in many cases hundreds of animals, to look at these trends across a large population. When taken together, looking at efficacy and toxicity, critical pieces of any drug development, drug discovery study, we're not just limited to the tumor or the heart itself. Other organs can be evaluated. Examples include the kidney. We can see blood flow here in the cortex of the kidney, looking at the liver itself and some of the major arteries and veins. We can look inside the colon, measuring wall thickness, similar thickness measurements done in skin. So again, if you're looking at the effects of your treatment, either from a therapeutic point of view or potential side effects, the vivo imaging systems can be utilized in a variety of different organs and disease states. A new area that we've been exploring is looking at the central nervous system, looking at brain imaging, everything from brain cancer, stroke, drug delivery involving the blood-brain barrier. This is all done using the vivo laser photoacoustic imaging platform. I've shown a picture of the system here today. I wanted to introduce this to you really for your information. We won't have specific examples with the vivo laser shown with our panelists today, but I did want to make you aware of this technology, which is really an add-on to our existing ultrasound technology. It's a photoacoustic technique that uses optical absorption with acoustic detection. So in the example of this transverse section of the adult mouse brain on the top left, this is all done using photoacoustics, looking at blood flow in the cortical region of the brain. And this is all done, again, using photoacoustics through the skull, all done in vivo, non-invasively, using the vivo laser photoacoustic system. To finish off, I just want to introduce the topic of data management. Once we've collected all of this valuable data, how can we streamline the management, the reporting, and all of the different workflows? We've recently partnered with In Vitro, and we'll hear more from Bill Capello today to talk more about the role of data management. This is a new tool that can take all of the visual sonic study data, measurements, and collate them all into reports, and it's a very powerful cloud-based tool that can be used for archiving, sharing data, and it's really a great way to manage all of this data and create, again, the reports and the outputs from the study that you're conducting, because ultimately it is the data that ultimately matters for these studies. So that gives you a good, I hope, overview of the technology itself, some of the areas where it can play a role in drug development, the role of data management, and I'd like now to pass things on to the other panelists to go into each of these different categories and different areas to expand and show you really some very good case studies of how the technology can be utilized.
Wonderful. Thank you, Andrew. And that's a good snapshot of what the vivo imaging systems, what the technology is on each of the vivo imaging systems, both the ultrasound and photoacoustics. We would like now to move on to our first speaker, who will be talking on the topic of drug safety in the context of cardiotoxicity. Jonathan Hand from Pfizer will be our next speaker, and we'll pass the privileges to him. Jonathan.
Yeah, thank you. Thank you very much. So thank you, everyone, for the opportunity to speak this morning. I appreciate it. So as Andrew highlighted, I'd like to share some of our experience in the drug safety realm using echocardiography to support our various projects over the years. And up front, these are very involved studies, so I'd just like to acknowledge and thank not only the team at Pfizer, but also some of the HESI Preclinical imaging members who we've worked together sort of working on various strategies to implement echocardiography, as well as the folks at Visual Sonics for a great collaboration over almost a decade now. So thank you to everyone up front. Today, what I'd like to cover in the next 10 minutes or so is really, it focuses oncology because that's what I work on a lot, but as mentioned previously, this spans multiple therapeutic areas. I'll give a little background how we got into using echo, what our basic image and analysis protocol entails. I'll do 2 very brief case studies and then just tie it up all at the end. So in the beginning, in terms of oncology, it's really been driven by this history of chemotherapeutic agents, namely doxorubicin or adriamycin, carrying with them a unforeseen cardiovascular side effect. And in the terms of doxorubicin, that was this long-term, non-reversible loss of ejection fraction and cardiovascular mortality that was complicated to treat, but was clearly dose-related, as you can see from the graph here at the bottom. The higher the dose in terms of mg per meter square you get in your lifetime, the higher probability of developing heart failure. And so this was the first time it was really observed that some of these chemotherapeutic agents may be having effects on other organs that may take months or years to manifest. And it was recommended that echo is a way to potentially monitor for these in the clinical setting. More recently, an anti-HER2 antibody, trastuzumab, also was found to have some cardiovascular liabilities. So it's well known that the HER2 or EGFR pathway is an oncogenic driver in breast cancer. And when you block this pathway, you can see very nice efficacy. But this pathway is also important in normal functioning adult myocytes, and blocking that pathway in these tissues can lead to typically what's seen a loss of functionality. So you lose ejection fraction 15 to 20% in these patients. Of concern to oncologists is that these 2 agents are sometimes used together, and indeed, as you might expect when they are, you see an increased incidence of cardiovascular side effects. So this really set our stage in interest in being able to detect these things early. And it's not just limited to these 2 agents, but in interest of time, that's all we'll cover today. The other thing that's happened is there's heightened awareness of these potential issues, not only with the clinicians and obviously with the pharmaceutical industry, but also with the regulatory agencies that approve these medicines, whether it's in the United States or Europe. And so for us in oncology, there's been emergence of a new field called cardio-oncology, which is, as you might expect, comprised of clinical and preclinical cardiologists and oncologists getting together and discussing this issue. And it's led to some nice outcomes in terms of guidance for ways to potentially control and understand cardiovascular risk with various oncology treatments. I would also like to point out that it's not just the field of oncology, but also diabetes or cardiovascular research, as well as anti-inflammatory research that also is under increased scrutiny for potential cardiovascular side effects. And one of the key things coming out of various meetings and collaborations is that the standard, clinical biomarkers of, say, troponin and BNP are not enough in isolation, and they strongly recommend other evaluations, more specifically echocardiography. And so we were very pleased to hear this, as it is something we have been doing preclinically for some time now. And what's laid out in this table, and I won't go through it in detail in the interest of time, but are various modalities that are used in the clinic and their pros and cons for various categories here down the left side. And all I'd like to point out here is echocardiography carries a lot of these advantages, not only in the clinic, but preclinically, and it's something we've been doing for over a decade now, and we see that as a key modality. And we see it that way for some of the same reasons that Andrew outlined previously. So it gives us the ability in our preclinical and clinical studies to evaluate structure and function simultaneously as well as over time, right? So we get that nice serial assessment of our structural and functional parameters. And then, of course, a big one for us is the translational opportunity the whole bitch to bedside approach, whether it's human to mouse or rat, or the reverse translation where we actually have some clinical data first, and we want to see what we can observe preclinically. In addition to that, I think there's some very nice emerging techniques that I won't cover today, but such as strain, that really have the potential to increase our sensitivity, not only clinically, but preclinically as well. And I'd also like to mention that echo is an important piece of an overall integrated approach that we take here, where we use all our preclinical data. So we typically have blood pressure, heart rate data, ECG data, as well as histopathology in terms of cardiovascular structure and function. And we look at all that data together to form a full risk assessment. How do we do this? Well, outlined here is our basic protocol for evaluating the left ventricle in a mouse or rat. We do a long axis view, again, peristernal long axis, as well as peristernal short axis at the level of the papillary muscles. That's number 1 and 3 here. And from both of those, we may or may not, depending on the study, take a long axis or short axis in mode and associated measures with that. And we also take an apical 4-chamber view, both the view itself as well as a mitral Doppler flow pattern as well. And that's it. In the mouse or rat, this takes us around 10 minutes or so per animal, so it's pretty high throughput for us in terms of a technique, and allows us to get a lot of information with hopefully as minimal time investment as possible. From those images, what we're interested in are a few different parameters listed here. So in terms of structure, of course, wall thickness, ventricular mass, and chamber volumes and dimensions. And what we're trying to do is go from needing to excise the heart to see hypertrophy to being able to detect that with echo techniques shown here on the M mode, where you clearly can detect hypertrophy over time. In terms of LV function, the key parameters, as you might expect, in systolic volume, cardiac output, ejection fraction, fractional shortening. And again, we can track all those over time. In terms of diastolic function, which we are also very interested in, the relaxation of the heart, we typically, our basic protocol, as I mentioned, is the Doppler outflow pattern. Shown here is the normal E to A ratio. On the right is that hypertrophic ratio that you might expect in a larger heart. But in addition to that, we do, on occasion, also employ the tissue Doppler techniques of the vivo 2100 and do myocardial tissue Doppler at the level of the mitral annulus and get that typical pattern shown here in the middle of the E prime, A prime, as well as those isovolumetric relaxation and contraction times. So that is our basic workup. Of course, there's many capabilities of the vivo 2100, and we certainly do customize based on what we're looking for, what we're interested in, left versus right heart, Doppler, outflow, et cetera. So lots of options there that can be covered. In the next 2 slides, I'd like to briefly cover just 2 case examples. This one is taken from a project that had some off-target pharmacology that was known to have a cardiovascular risk. So we ran a very simple acute study where we did a baseline echo and then treated animals either with vehicle or a dose response of our compound of interest. The example here is at 10 mg per kg. And so we did baseline echoes 1 hour post-dose, and what you hopefully can clearly see from the M-mode images here is that compared to baseline, we see a nice robust increase in contraction. And this compound was indeed a positive inotrope with increasing contractility in these animals, whether in the form of ejection fraction, fractional shortening, or other left ventricular systolic functional readouts. And this was happening at or near concentrations that were predicted to be efficacious, representing a clear risk for this compound. And thus, this compound was deprioritized, and other potential compounds in that series were explored. So that's one example of decision-making in terms of using ultrasound. And then I'll just finish up today with our classic study. This is over 10 years old now, and it's really just doxorubicin in the rat model. And this was a dose-response study of 1, 15, and 25 mg per kg doxorubicin given twice weekly over 2 weeks. And what I show here is ejection fraction, where we see a nice dose-dependent decrease or loss of function over time in these animals, most notably on day 14, where we see a 13 and 21% decrease in ejection fraction compared to baseline. And this matches up with the classic histology, the vacuolization observed in the rat hearts upon treatment with doxorubicin. And this was really the study that convinced us preclinically that you can get useful information from these models and potentially identify cardiovascular risks early rather than after you progress to the clinic. And it is indeed a useful tool. So just in closing today, some of the highlights I covered, there is increased awareness both in terms of regulatory agencies but also within the pharmaceutical industry itself of potential cardiovascular risks, and we're always working to try to mitigate these. And to do that, we have implemented more stringent preclinical and clinical monitoring for cardiovascular effects, and we feel we get the most bang for our buck using ultrasound techniques, particularly the vivo 2100 in the rat and the mouse. It gives us really valuable data in terms of decision-making as well as risk assessment. And finally, in terms of some of the new technologies coming on, as I mentioned previously, strain, we think these are going to continue to improve our capability to do this and are going to be key for us moving forward. With that, I'd turn it back over to Mina. Thank you.
Jonathan, that was great. That's a good snapshot on cardiotoxicity and toxicity in general. Its value right now in drug development and drug discovery. And Jonathan's expertise as pro shed some light as to how you would use it and perhaps the challenges, overcome the challenges that he has as he presented today. So we'll move on quickly to our next presenter who's going to talk on the topic of efficacy, Alexandra Acton from Regeneron Pharmaceuticals. I have a biography up there while we transfer the privileges to Alexandra.
Okay, thank you very much. I'll be telling you about our efforts where we utilize non-invasive imaging modalities, and that includes the micro-ultrasound, to assess the effects of VEGF blockade in tumors. Since not everybody is an expert on angiogenesis, I thought I'd start out with a bit of background. So during the development and growth of a tumor, the nearby blood vessels initially provide oxygen and nutrients and remove waste products from the tumor site. But then once a tumor reaches a certain size, the surrounding blood vessels will not be sufficient anymore, and more nutrients and oxygen is needed. And when this happens, the tumor releases factors that promote the growth of these new blood vessels, and this process is called angiogenesis. In solid tumors, the occurrence of angiogenesis is a mandatory event to support the tumor growth. The most important angiogenesis promoting factor known is vascular endothelial growth factor, also called VEGF. And when VEGF is released, it causes the endothelial cells, which compose the blood vessels, to divide and migrate into the tumor and form a new blood vessel network. So VEGF can bind to and signal through two different receptors, which are called VEGF receptor 1 or VEGFR1 and VEGFR2. At Regeneron, we've developed a fusion protein called VEGF trap or aflibercept, which is composed of the VEGF binding domains derived from VEGF receptor 1 and receptor 2 fused to a IgG1 Fc portion. So the way aflibercept works is by binding VEGF and as well as PLGF, placental growth factor, and VEGF-B, and thereby preventing them from binding to and signaling through their receptors, and that inhibits angiogenesis. VEGF blockade using aflibercept inhibits tumor angiogenesis and tumor growth in preclinical as well as clinical settings where aflibercept in combination with chemotherapy is currently approved to treat metastatic colorectal cancer following progression on oxaliplatin-containing regimens. As I mentioned before, VEGF blockade results in inhibition of tumor angiogenesis and delayed tumor growth. However, not all tumors respond equally well to anti-VEGF therapy. The identification of early response biomarkers that correlate with long-term efficacy could help identify tumors most likely to respond and thereby help select patient populations that would benefit. So what we did is we assessed early molecular and morphologic and functional vascular responses following a treatment with aflibercept to determine which parameters could assist in predicting these long-term tumor growth response effects. So for those efforts, we utilized tumor models with a range of responses to VEGF blockade, specifically the sensitive Colo-205 tumors, moderately responsive C6 tumors, and completely resistant HT1080 tumors. In these three tumor models, we assessed vessel density changes at 8, 24, and 72 hours post-aflibercept treatment, and this was done by CD31 immunohistochemistry. As is shown by these representative images of tumors treated for 24 hours, as well as the quantitative assessment of vessel density over time in response to aflibercept below the images, a decrease in vessel density occurred in all three tumor types. So this suggests that the early changes in vessel density do not correspond to long-term tumor growth response and are not predictive, since we also see them in the completely resistant HT1080 tumors. So in addition to morphological changes, we were also interested in functional changes of the vasculature after aflibercept treatment, such as changes in vessel perfusion. As you probably know, the resolution limit of micro-ultrasound is at 30 micron, but this resolution limit can be overcome by the use of contrast agent microbubbles, which allows us the assessment of smaller vessels, and that's quite important when you look at tumors. So microbubbles are gas-filled lipid-shelled spheres with a diameter of 2 to 3 micron, and they're similar in size and behavior to an erythrocyte when they're traveling in the bloodstream, and they can be easily detected in the bloodstream due to their gaseous core and the strong reflection of sound. So to assess perfusion, we acquire images prior, during, and after contrast agent administration, and that results in a time intensity curve. The ratio between the steady state level and the baseline intensity is calculated, and that reflects the relative blood volume as a readout for perfusion. Thus, the use of microbubbles allows the visualization of microcirculation and quantification of perfusion. So utilizing this contrast-enhanced micro-ultrasound technology, we determined the perfusion of the three different tumor types that I've mentioned before when they were treated with control agent or aflibercept for 24 hours. And we observed a decrease in perfusion after aflibercept treatment in the sensitive Colo-205 tumors and the moderately responsive C6 tumors, but not in the aflibercept-resistant HT1080 tumors. These data were actually confirmed by other methods, which I don't have the time to go into, but all these methods show that early changes in perfusion correspond to long-term tumor growth response, suggesting that perfusion changes could be used as an early predictor of treatment efficacy. To assess if early perfusion changes reliably predict long-term tumor growth inhibition, we extended our efforts to additional tumors. So we tested a total of 6 tumors for microvessel density changes and 8 tumors for early perfusion changes and correlated the results with long-term tumor growth inhibition. The correlation between long-term tumor growth inhibition and perfusion, shown in the bottom graph, was very good, as indicated by an R-square value of 0.73, with an R-square value of 1 being a perfect correlation. However, the correlation between the long-term tumor growth inhibition and microvessel density shown in the top graph was pretty poor, with an R-square value of 0.09. So taken together, the data I showed you indicate that early changes in perfusion of aflibercept-treated tumors is predictive of long-term tumor growth inhibition in a variety of preclinical tumor models tested. So just to summarize, we studied 3 different tumor xenografts with different responses to aflibercept and aflibercept was able to decrease vessel density in all 3 tumors. However, early perfusion changes correlated with tumor growth inhibition, and we only saw the perfusion decreased in tumors that are responsive to aflibercept, while perfusion did not change in the resistant tumors. And this decreased perfusion resulted in hypoxia in responsive tumors, while no hypoxia was observed in aflibercept-resistant tumors, kind of as an inverse correlation to perfusion. The efforts were extended to additional tumors, and we found that early perfusion changes, but not early vessel density changes, correlate with long-term tumor growth inhibition. And this, taken together, this suggests that contrast-enhanced micro-ultrasound imaging allowed us to identify perfusion changes as a predictor of aflibercept response in xenograft models. And I would like to thank everybody in the oncology and angiogenesis team at Regeneron, especially Alex Adler, who is one of the main people who's done the ultrasound imaging here. And then I'd like to thank everybody in the Visual Sonics team for a very, very nice collaboration that's been going on for a couple of years now. Okay, that's it. Thank you. I'll turn it back over to Mina.
Thank you, Alexandra. Wonderful presentation. Just adds on to Andrew's talk on the concept of tumor volume, then bringing it to the level of angiogenesis using looking at blood flow, whether it be power Doppler. But now Alexandra has expanded on that and looked at the microvasculature, looking at the microenvironment. And most of you in the field of tumor, in the field of oncology, you know the changes, specifically angiogenesis and hypoxia being a menace to treatment today, and it's causing a major challenge to standard of care. So it's great to see how the micro-ultrasound system or micro-ultrasound technology can alleviate some of those challenges early on in your preclinical strategy and enhance your preclinical program to better bring a drug so patients can benefit. So thank you very much, Alexandra. We will now move on to another obstacle in these kinds of longitudinal studies with large sample size, which is data management. And I'm pleased to introduce our partner in crime, William Kirk from In Vitro. He's going to talk about, he's going to introduce the Visual Sonics IPACS study management solution. So, Bill, it's all yours.
Great. Thanks, Mina, and thanks, everyone, for attending today and for WPC for hosting this webinar. I'll take a few moments to introduce the Visual Sonics IPACS product that we're jointly launching between In Vitro and Visual Sonics this year. But first, I'll spend a few moments just to introduce everyone on the call to In Vitro as a company, to give a little perspective of our background and why we're partnering with Visual Sonics. And then we'll go into the product itself and how it's, we're hoping, adds value to sonographers that have challenges both in pharma and academia for managing the data coming off of the Visual Sonics instruments. So In Vitro as a company, we position ourselves as an imaging solutions provider. We're agnostic to an imaging technology, although we have our roots in PET, SPECT, MRCT, have really branched to many of the modalities that groups work with both in vivo and ex vivo in assessing drugs and therapeutic programs in the preclinical and translational clinical space. We're based in Boston. We have offices across the U.S. and Europe. And unlike other software groups, we have a team of over 50 research scientists that are devoted to executing and running imaging trials ourselves, as we have a contract research group that will execute preclinical to translational first-in-human clinical trials across many of the modalities I just previously mentioned. Our main focus as a company, however, is on data processing and data image informatics solutions, both as a service and as a software product. And we'll be focusing most of the conversation today on our software platform, that being the Visual Sonics IPACS. The last thing I'll just note in terms of our company as a whole is the breadth of work that we do in the pharmaceutical and academic space in supporting scientific research using imaging. You see some of the stats in the top two sections for contract research in our in-house image analysis group that processes and analyzes data, is that we develop our software platform not just for external use, but also for internal use as well. As if you're measuring up against other core laboratories, both in pharma and academic groups, we're in the top 5 in the world in terms of volume of studies and volume of data that come through our group. And so we deal with, on a daily basis, the challenges of high data volume, data management, and data sharing to facilitate information being accessible to program leaders to make decisions of moving a program forward or not. In terms of where our software platform is as a whole in the marketplace, as I mentioned, we're modality agnostic, although we do develop solutions specific for a imaging instrument vendor's particular modality and application. For instance, like we are partnering with Visual Sonics. But in terms of where we are in the market, we're in over 150 laboratories worldwide and adopted by over 70% of the top pharmaceutical companies for utilizing our solutions both as a data management and viewing and analysis solution across their in vivo and ex vivo imaging data. As Andrew highlighted in the beginning of this talk, the Visual Sonics IPAX is a new and innovative data informatics solution for specifically Visual Sonics users. We've been collaborating over the last year to develop a solution that leverages our overall IPAX, which is our data management platform, with data coming off of the vivo systems to offer solutions to Visual Sonics users to organize and make their data more accessible across the team, to streamline the data mining and reporting workflows, as well as to offer solutions that make data more accessible into a web-based platform, as well as more accessible into how that data is organized into reports for QC or presentation purposes. I'll start with just a little background for how data is generated in the Vivo Lab software platform, which is the local data management, viewing, and analysis solution that comes with every Visual Sonics instrument. As you see here, they have a study browser which organizes local data into a study series image-centric management display. And the current data management support, again, it's local and specific to a defined pool of data. Where the IPAX adds value is it can offer more efficient and streamlined data management tools built specifically for how ultrasound Visual Sonics users will need to interact with that data. As what has been pointed out by the previous two speakers, there's a wealth of information that comes out of the image data from the Vivo Lab software, coming from the data off of the Vivo systems. And all of this data needs to be organized in a more systematic and logical order so that users can go back in time and pull their data according to how they want to interact with that data, from organizing into specific spreadsheets to organizing into presentation-ready material. And so all of the data generated out of the Vivo Lab system, the image data, the associated metadata, so image acquisition information, as well as any of the analysis output, can be stored into the IPAX in a synchronized manner. So for those that are using the current reporting engine within the Vivo Lab, you're familiar with some of these templates that I'm displaying, although the font's a little small here for the image render. But in essence, you have per series, per study, an aggregation of really all analysis specific to that given series. And so if a user wanted to manipulate how this data was displayed or specifically find a unique measurement or calculation data point, that can become challenging very quickly. And so what we have here is a solution within the IPAX platform, what we're labeling as the Visual Sonics IPAX, which allows users to access their data through the web in a web browser or other terminal access means. But more importantly, the data is now centralized and accessible to the entire team in real time. So as data is generated off of the instrument, it then can be made available to people working outside of the lab, either remotely or at their desk, and then take advantage of the powerful data mining and reporting capabilities within the IPACS framework. And so some examples of how the data looks organized within the IPACS, you'll see here that this is a very similar view to how the data is displayed in the study browser view within the Vivo Lab software. And we do that so that there is similarity to how the users interact with the data both in the Vivo Lab software as well as in the IPACS. But also what's nice is that you'll see we're mapping data according to a DICOM-like standard, and we're involved in a lot of efforts to standardize how preclinical data is managed, both with ultrasound data, but also with all modalities that, whether that data is in the form of DICOM or in non-DICOM data, so that researchers can quickly find a data set, whether it be an ultrasound image or they want to map directly and find the correlated histopathology data or PET, SPECT, CT, and whatever complement modality you're using to assess that drug or therapy. By having the data available, again, in the database of the IPACS, users can filter on a pool of data to reduce to a specific set of the data that they're interested in reporting on. Here, just a simple example of reducing the population of data to cardiology data, and then walking through a wizard-like pipeline for how a user would generate a report outputting measurement and calculation data points of interest. So for instance here, if I go back, it's taking a current product offers the ability to take all measurement and calculation data available within the pool of data you see here. And then you can, as a user, selectively manage which data points are then aggregated into a spreadsheet. And this is what we've done so far is really only the tip of the iceberg in terms of the report templates, as we offer default report templates as well as offer customized report templates capabilities to the end users, so that if you're trying to reproduce what you're generating manually by copying and pasting from spreadsheets generated out of the Vivo Lab to what your final output spreadsheet needs to look like for, again, QC or reporting, copying into PowerPoint presentations, this should all reduce the time spent to a matter of seconds. We have been doing some interesting case studies with groups, and we're reducing time spent on managing data from days to minutes, really, in terms of leveraging the already existing data management structure of the IPACS with what has been built specifically for Visual Sonics users. And then this output is then exportable into a spreadsheet form as well as into a PDF output form, which allows us to embed other objects such as screen captures, as well as manipulate tabular and graphical data to the user's liking. And so we're just really getting started with this product, but we're looking forward to supporting more use cases in the cardiovascular space as well as outside of the cardiovascular space into some of the other therapeutic areas of research that are utilizing the Visual Sonics instrumentation platform. So with that, I know we're close to the 12 o'clock time. I just want to take a moment to thank, again, the Visual Sonics team for all the great work we've been doing and all the beta customers that have been helped develop this product to where it is today. And then, obviously, to WPC for hosting today's webinar. Mina, I'll turn it back to you.
All right, thank you, Bill. That should have shed light to those of you who are using Excel. I noticed that we did ask the question as to what data management tool you're using. You can see these are in some fashion similar to an Excel report, but it's much easier, much more streamlined to triage only the data sets that you want to see from this huge pile of data sets, so you can data-rich study from the Vivo imaging systems. So on that note, there is a question, and we are progressing to the question section of this symposium. I'm taking a couple of questions in the interest of time. Question for you, Bill. The last part of your talk, you talked about quality control. Now, for the For the purpose of the pharma segment and those of us working in drug discovery and development, while quality control is important, there is also the GLP, compliance to GLP, that's very important.
Yeah.
Now, how does one identify that kind of potential within the Visual Sonics IPACs or the IPACs environment?
That's a great question and has come up already in some of the few conversations we've had with some of your customers in pharma. Our platform does scale to a GLP compliant solution. It both supports 21 CFR Part 11 compliance as well as pharma-specific GXP compliant workflows. And I emphasize it in that way because in our experience when we were originally validated by Merck a few years ago to have the first iteration of our GXP compliant solution that manages all of their histology through clinical data in the IPACs for archiving purposes, it's not only a software solution, but a total solution around the back-end storage solutions as well as the connection points of data coming in and out of the IPAC software. But we have a compliance team that if someone needed to scale the Visual Sonics IPACs to support compliant workflows for to support drug filings or archiving in compliant manners, our team can support that.
Fantastic. I was a question for Alexandra. The question states that the contrast-enhanced ultrasound helped with the prediction of the efficacy of the drug. Now, where does that drug stand today in terms of its benefits to patients, in terms of timing? Would you be able to comment on that?
In one of my initial slides, I mentioned that aflibercept is currently approved for metastatic colorectal cancer, and that's where that stands at this point.
Okay, so it is in the clinic for, is this a metastatic?
Metastatic colorectal cancer for patients that have progressed on oxaliplatin-based chemotherapy.
All right.
It's in combination with chemotherapy.
Okay, so it's in adjuvant. Thank you, Alexander. Mm-hmm. And here's the question for Jonathan with respect to strain. You did mention that strain has potential for expanded work in terms of drug safety. How do you conceive that is possible with drugs with respect to cardiotoxicity and using strain to address cardiotoxicity?
Yeah, great question. So I'd say we're in the validation phase of that, and so what we've been doing is taking sort of standard agents like doxorubicin or others. And really what you can do is take your standard parasternal long axis or short axis image, and as long as you collect at high enough frame rate, you can do that analysis offline, right? So that's the beauty of strain is the data is typically there, so it's something you're interested in, you can go back and get. So we're really in the beginning stages of analyzing if some of those strain and strain rate parameters are indeed more sensitive. And that's where the HESI collaboration has been really useful, and some of our colleagues at GSK are probably a little ahead of us in terms of validating that as a biomarker. But I'd say the preliminary data looks pretty good. We can see changes in strain and absence of changes of some of our conventional parameters, which is making us think that it is indeed a more sensitive technique.
Oh, fantastic, Jonathan. So we look forward to those reports. And also, Alexandra's publications are out there. We will be able to share that with you. I'll provide you with some key resources that you have on our website that you can use to your advantage to make sure you have the latest and the greatest insights to drug safety and efficacy. The very last question is with respect to application. We had a question whether CSF can be visualized in the choroid plexus. We're talking about the brain, and this, I think, comes from the aspect of using photoacoustics. While I don't have enough information, so I'm assuming it's either in the embryonic stage. This can be done, and we would like to actually, before I say something, I'd like to actually bring in our neurobiologist, a specialist in this field, to your attention, and we will address that offline. So in the interest of time, we will close off the questions period, and all other questions will be addressed offline. Just to close off the symposium, we encourage you to stay connected to innovation. Here are your key resources that you will see in the next slide. You can either request for bibliography at our website, www.visualsonics.com. Keep yourself updated with the Vivo News, and to sign up the Vivo News, just check in our homepage to register for this newsletter. You can also sign up for automatic alerts or updates through the Vivo Journal Club. also available resource on the Visual Sonics website. And you can see the URL right here. We also would like to bring to your attention, as we like to meet you, and if you want to meet the team face-to-face as compared to our cyber visit today, we will be at World Preclinical Congress, June 10th to 12th. You can meet our team that specializes in the drug safety and efficacy field. Finally, we don't stop there. We continue to educate you on the resources, more on a webinar that's coming up in June 10th at 1 p.m. It's an on-demand tutorial. We will walk you through some latest and greatest software that helps you advance your research. And this time, the topic is hearing in color, visualize and quantify molecular markers with photoacoustic imaging. You heard briefly about photoacoustic imaging in Andrew's presentation today. Here's another Andrew Heinmiller, Product Manager of Photoacoustics, will be presenting a tutorial on June 10th. This brings me to conclude this symposium. I really would like to appreciate the time taken by our guest speakers for sharing their expertise and enriching you with objectives that are pertinent to drug discovery and development. And I'd like to thank CHI and WPC for giving us this platform. Thank you, Elizabeth.
You're very welcome, Meena. Thank you to everyone who came and spent some time with us today. We look forward to seeing you at future events. Have a great day.